going to tell you that what is metabolism in biochemistry. So just starts with the brief description of about the biochemistry. So see, biochemistry is a branch of medical science in which we deals with the chemical reactions which are happening in the living body or living organisms. So those chemical reactions which takes place inside the living organisms are comes under biochemistry. So when we are talking about the chemical reactions which takes place in the living organisms, that is known as biochemistry. So in biochemistry, you have already learned that there are the biomolecules like carbohydrates, proteins, lipids, nucleic acids, and also you have read the water balance, acid-base balance, right? So in today we will discuss about the metabolism of your biomolecules, like that how carbohydrates are metabolized in the body and how energy is synthesized from these biomolecules. So let's start with the today's session that is on the overview of biochemistry metabolism. First of all, I want to tell you that what is metabolism. As you all are aware that biochemistry is a branch of science that deals with the chemical reactions happens in the living organisms. And the meaning of the metabolism is that when something or when a network of chemical process happens in the living body to maintain life, then this refers as metabolism, right? Whenever a number of or a network of chemical reactions takes place in the living body that are necessary for the maintenance of the life, that is necessary for the living organisms. Without the reactions, the life is not possible. That is known as metabolism, right? Now comes to the bifurcation of metabolism. Metabolism is a term which is collectively made up of catabolism and anabolism. Catabolism is, it means the breakdown of products to produce energy, right? So metabolism is collectively made up of two terms, catabolism and the anabolism. Catabolism is breakdown of the biomolecules to produce energy, and on the other hand, anabolism is use of the energy to make new products, right? Catabolism is the formation of energy from by breakdown of the products, and anabolism is from the energy synthesize new structures, new proteins, new carbohydrates for the body. So collectively, both of the reactions takes place simultaneously in the body, and collectively they are known as metabolism, which is necessary for the maintenance of life, right? Next is the energy metabolism. So the energy metabolism means the way that the body obtains and spends energy from food, that how the body obtains the energy from food and how the body spends the energy which is derived from the food. That is known as energy metabolism, that how the energy is made from the food and how the energy is spent on the body to form new things like new proteins, new lipids, new carbohydrates to maintain the life in a living manner. So as now the catabolism and the anabolism, as I told you that the anabolism is the building up of compounds from small molecules into larger one. Energy is used for this process to take place. Whenever anabolism takes place inside the body, so what happens? Small molecules are collectively combined to form a large molecule, and in this process energy is used, right? Energy is not liberated. In this, energy is used. Next is the catabolism. In the catabolism, the breakdown of molecules, the larger molecules will break into the smaller units and the energy is released, and this released energy is further undergo in the anabolism and maintain the metabolism, right? For example, glucose catabolism in the release of CO2 and water. Whenever you intake oxygen, the glucose which is present inside your cell will break down and produce ATP as well as carbon dioxide and the water. And finally, the CO2 is exhaled by the lungs and ATP is used by the cell for the cell division, cell growth. All right. let's talk about the energy, that is ATP, adenosine triphosphate. This is the main energy source for the cells, as we are talking about again and again, energy, energy, energy. So what is the energy? This is the ATP. It is the main or major source of energy for our cells, right? It is used for the muscular contractions. The contractions of muscles is also because of the ATP. The enzymes activity, the enzymes which perform various biochemical reactions inside our body, they are also energy dependent. Without energy, the enzymes are not able to perform their chemical reactions. Catabolism result in the production of many ATP molecules, which ultimately gives what? Energy, right? Used by the body when energy is needed. ATP molecules are used by the body when the energy is required. So ATP is produced and it fulfills the energy requirement of the body. All right. Hydrolysis breaks the bond in ATP, thus releasing energy. First of all, glucose enters the cell. Glucose will break down in the ATP, CO2 and water. Then the ATP is hydrolyzed and it liberates the energy. So that's why it is also known as the major source of energy, right? Cells are dependent on ATP for fulfilling or performing their actual functions they have. Next is the metabolic efficiency. So what is metabolic efficiency? It means that how much energy is synthesized from the food and how much energy is used. So food energy is converted to ATP with approximately 50% efficiency. It means the food we eat, the food is digested, absorbed, and then it is converted into what? It is converted into ATP with approx 50% efficiency. The other 50% is released as heat and the remaining 50% is released as the heat. Now the cell, because the cell is the major site for the synthesis of the activity and for the energy production, and because also it is a site for metabolic activity. So as we are talking about the cells, so out of the every cells or of the body, the liver cells are most metabolically active. If I'm talking about the cells, so above all the cells, the liver cells are more metabolically active because most of the chemical reactions and most of the things are synthesized in the liver cells. How energy is produced? Basically, see, we are talking about the biochemistry metabolism. So when we are talking about metabolism, it's all about what? It's all about the energy. So next is that how energy is produced inside the body. So there are three stages to for the production of the energy. Number one, like your biomolecules like proteins, carbs and fats are broken down during digestion, and finally they are absorbed into the smaller units like amino acids, monosaccharides and fatty acids respectively. For example, the meaning of that point is that you are eating the complex food which contains proteins, carbohydrates, fats in their complex form. So our body is unable to absorb the complex form of the protein, carbohydrates and fat. To absorb the biomolecules needs to be broken down into smaller units, and this is known as digestion. When we eat the food, it will goes inside the stomach and then ultimately in the intestine, and here the enzymes will act upon the food and convert the food into smaller units, or we can say that in the basic units which can't further hydrolyzed. Once the food is broken down into the smaller units, then it is absorption ready. It is ready to absorb. So what is the simplest form of protein? Simplest form of protein is amino acids. To absorb the protein in the body, it must be converted into its simplest form, and that is amino acids. Next is carbohydrates. Carbohydrates are the sugars. To absorb the carbohydrates in the body, it must be converted into the smaller form or the simplest form, that is monosaccharides. Next is the fat. Fat broken down into the smallest unit, that is fatty acids. So these are the smaller or simplest units in which our body are able to absorb. So first stage is about breakdown of complex food into smaller units, and then smaller units it gets absorbed by the body, by the intestine, and ultimately it is moved towards the peripheral blood, or it is moved in the blood. Then what happens in the second step? In second step, these smaller compounds are further broken down into two carbon compounds, right? Then after absorption, the basic units are further broke into carbon compounds. Number 3, compounds are degraded into CO2 and H2O. So when the compounds are degraded into carbon dioxide and water, then side by side there is the release of energy. What are the helpers in the reactions? We have helpers are enzymes, coenzymes. So basically what are enzymes? They are the biocatalyst. Basically they are made up of proteins. They are necessary for catalyzation of the chemical reactions without being changed in the process. They are not getting changed in the process. They just catalyze the chemical reactions. Example, protein catalyst. For example, protein is broken down into simple form by the trypsin. So trypsin is an enzyme. It is a biocatalyst. Coenzymes. Coenzymes are the additional material which is required by the enzyme to perform its activity, right? They help the enzymes to perform its appropriate activity. All right. Next is breakdown of nutrients for energy. That how the nutrients will break down for the production of energy. Number 1, glucose breakdown. Number 2, glycerol and fatty acids breakdown. Number 3, amino acids breakdown. Because as I told you that in the food there is the carbohydrates, protein and the lipids. So to generate energy, the breakdown of glucose, fatty acids and amino acids take place to produce energy. See, fats, carbs, proteins undergoes a common pathway which we know as the metabolic pathway and leads to the production of energy. Let's see one by one. Number 1 is glucose breakdown. So glucose is breakdown by a process which is known as glycolysis. Glyco means glucose, lysis means breakdown. So a reaction in which glucose is breakdown or glucose is degraded to produce pyruvate with the production of 2 ATPs. It is 2 type of cycle. It takes place in aerobic conditions as well as in anaerobic condition. In aerobic condition, the end product is pyruvate, and in anaerobic condition, the end product is lactic acid. So when there is an availability of oxygen, the pyruvate is converted into lactic acid, and if the proper oxygen is available, the pyruvate is converted into acetyl coenzyme. So what is glycolysis? Conversion of glucose, or we can say that degradation of glucose into pyruvate with the production of 2 ATPs. If the oxygen is available, then it will lead to the formation of acetyl coenzyme. If oxygen is not available or less available, then it leads to the production of lactic acid. The pathways of pyruvate and acetyl coenzyme. See, once the glucose is degraded, it will form pyruvate, and from pyruvate leads to the production of acetyl coenzyme. This acetyl coenzyme can also lead to the production of amino acids and also leads to the production of fatty acids, and also it will go inside the TCA cycle, that is known as tricarboxylic acid cycle, for the production of energy. See how the acetyl coenzyme is further breakdown in the TCA cycle to produce energy. See, there is a systematic steps of tricarboxylic acid cycle. Acetyl coenzyme is finally converted into the ATPs. Then these ATPs finally goes into the electron transport chain, and they finally enters the cycle and leads to the production of what? Leads to the production of energy. Okay? The resultant acetyl coenzyme, it splits with the help of carbon dioxide and oxygen, with the help of carbon and oxygen, and it is stored ultimately in the form of ATP, right? And this ATP is now available for the body's use. Next is glycerol and fatty acid breakdown. Triglycerides, if I'm talking about the fatty acids, it means the simplest form is triacylglycerides or triglycerides. They are broken down into glycerol and fatty acids, which is known as lipolysis, means breakdown of lipids. So once the glucose is converted into pyruvate, the glycerol are also breakdown, right? And the breakdown of glycerol leads to the production of glycerol and the fatty acids. Glycerol will help in the conversion of glucose to pyruvate, and fatty acids help in the production of acetyl coenzyme. Next is amino acid breakdown. That how the amino acids will break. See, glucose is converted into pyruvate, pyruvate into acetyl coenzyme, and acetyl coenzyme is finally goes into the tricarboxylic acid cycle, and these steps are facilitated by the amino acids. So amino acids will help in the conversion of glucose to the acetyl coenzyme, and finally the acetyl coenzyme goes into the tricarboxylic acid cycle, and here the amino acids will support the conversion of acetyl coenzyme into adenosine triphosphate. Right. So the amino acid breakdown goes in several steps. Like first one is deamination. In deamination, the amino acids is break down into keto acids as well as in the ammonia. See, ammonia is the harmful product for the body. So the ammonia will toxic to our body, so it needs to be converted into non-toxic product. So it is performed by the liver, which is known as transamination. In transamination, the ammonia is converted into urea, right? This is the end product of protein metabolism, right? In transamination, the ammonia is converted into urea in the liver, and this is known as urea cycle. And finally, the urea, the non-toxic waste product, which is the end product of protein metabolism, is excreted via the kidneys with the help of urine, right? So what is needed for the urea excretion? The urea is diluted with the help of water, and it is ultimately goes out from the body in the form of urine. See the TCA cycle, because ultimately goal is to enter the acetyl coenzyme into the tricarboxylic acid cycle to produce energy. So what is the function of tricarboxylic acid cycle? The basic function of the TCA cycle, Krebs cycle or citric acid cycle. See, TCA cycle has three names: tricarboxylic acid cycle, Krebs cycle, and your CAC, citric acid cycle. So the function is to convert the acetyl coenzyme to carbon dioxide to produce energy. Number one, oxaloacetate combined with acetyl coenzyme to begin the cycle. To begin the cycle, number one, oxaloacetate needs to combine with acetyl coenzyme, and the resultant is the production of ATP. Now moves towards the electron transport chain, because the electron transport chain is the primary site for the synthesis of ATP. So it uses oxygen to convert the products of TCA cycle into energy. See, the end products of the TCA cycle with the help of oxygen is converted into the ATP with the help of electron transport transport chain, right? The electrons that bound, it is an important concept to remember is that an electron is not a fixed amount of energy. The electrons that bonds the hydrogen to the vitamin B complex have a relatively large amount of energy. In the series of reactions that follow, they lose the energy in small amount units. At the end of electrons with their hydrogen, they are attached to the oxygen to form what? To form water. In some of the steps, the energy the electrons lose is captured into the ATP in coupled reactions, right? For example, coenzymes carries the hydrogen ions. The hydrogen with ions having the lower energy electrons, hydrogens with the lower energy electrons combine with oxygen and they ultimately form water. Energy is captured in the ATP in each and every step. Why is fat in higher energy? See, as I'm talking about each, as we talk about the biomolecules, so the maximum amount of energy is produced by the fat. Why? Because fats carbon bonds are easily oxidized and yields high energy or ATP. One glucose molecule produces 38 ATPs when oxidized. So once one fatty acid which is composed of 16 carbons will give 129 ATPs when oxidized. See, what is the formula of glucose? The formula of glucose is C2 C2H6O2, right? C6H2O6. So here is the 2 carbon, and just wait. See, when we see the chemical formula of glucose, that is C6H12O6, right? If one glucose gives 38 ATP molecules when oxidized, then a fatty acids, if you are seeing this structure, the glucose is having 1, 2, 3, 4, 5, 6 carbons, right? But a fatty acids have 16 carbons. So if one glucose that is arise from 6 carbons gives 38 ATP molecules, then one ATP or one fatty acids gives 16 carbons. That produces 129 ATPs when oxidized. That's why the fat have the higher energy. Now the unit is about the organ function test. See, in your biochemistry metabolism subject, the unit 1 and 2 is about, unit 1, 2, 3 is about the metabolism, that I already told you, that how the biomolecules are metabolized to form energy. And the unit 3 is about the organ function test, which is very important as a point of view of lab technician. You have to deal with the laboratory test, and that is your unit number 4, that is the organ function test. Number 1, we will discuss the organ function test one by one. Number 1 is liver function test, so which is also known as LFT. So what is LFT? LFT is generally performed to check the ability or functional capacity of the liver. Is the liver is function properly or not? So to check the functional capacity of the liver, we have to perform the LFT, right? In the LFT, there are some parameters like ALT, that is alanine aminotransferase, AST aspartate aminotransferase, ALP alkaline phosphatase, bilirubin, that is total bilirubin, direct bilirubin, indirect bilirubin, albumin, total protein, prothrombin time, gamma glutamyl transferase, that is GGT. So they are the parameters which comes under the report of LFT, right? Number 1 is ALT, that is the liver. So ALT, AST, ALP, GGT, they are the enzymes which is produced by the liver. They are the liver enzymes. So basically, they indicates the changes in the liver cells, right? So the normal range of ALT is 5 to 40 international unit per liter, and this enzyme indicate the liver cell damage. In case of liver damage, liver cells damage, the ALT levels will high than the normal. The normal range of AST is 5 to 45 IU per liter, and if the value go above the normal, then it reflects the liver damage. Next is the ALP. The normal range of ALP is about 40 to 115 international unit per liter. The value above 115 international unit per liter is elevated and is shown in bile duct obstruction. If there is an obstruction in the bile duct, then it will lead to the elevations of ALP levels in the LFT parameter. Next is the bilirubin, that is total bilirubin, direct bilirubin, indirect bilirubin. So basically, if any of the bilirubin gets high in the blood, then it's indicate the effect in the excretory function of the liver. The normal range of the total bilirubin is your 0.2 to 1 mg per dl. The direct bilirubin is 0.2 to 0.6, 0.2 to 0.4 mg per dl, and the normal value of indirect bilirubin is 0.2 to 0.6 milligram per dl. Next is the albumin. Albumin is the protein which is synthesized by the liver. If there is a decreased concentration of albumin shown in the blood, it means the liver is inable or incapable to synthesize the albumin. So the normal range is albumin is 3.5 to 5.0 gram per deciliter. Next is the prothrombin time. See, prothrombin time is a test of clotting factor or clotting cascade, as the prothrombin is synthesized by the liver, which is necessary for the coagulation of the blood. So it take as a part of the function of the liver, right? It doesn't come in the LFT parameters, but it indicates the function of the liver because it is synthesized by the liver. If PT is high or increased, then it is indicate the deficiency in the liver also, that liver is not able to synthesize the clotting factors properly. That's why the blood is not clotting. Next is the GGT, that is also a liver enzyme. It is a marker of alcohol related or bile duct issues. If the patient has alcoholic liver, fatty liver, then the GGT is high than the normal, and the normal range of GGT is 0 to 40 international unit per liter. If it goes above the GGT, then it indicates that the patient is suffering from alcoholic liver. Next is about the kidney function test, right? So as LFT, the kidney function test is also ordered to a patient or ordered to a person if he is suffering from any symptoms which shows that maybe the kidney is affected. So to check the functional capacity of the kidney, we have to perform the kidney function test over the patients. The parameter which comes under KFT is serum creatinine, blood urea nitrogen, estimated GFR, urine analysis, creatinine clearance, electrolyte panel, and imaging ultrasound. Just wait for 2 minutes. Thank you for the patience. So number one is serum creatinine. Serum creatinine is a key marker, or we can say that specific marker for kidney filtration efficiency. Next is the normal range of creatinine is 0.5 to 1.2 milligram per deciliter. Next is blood urea nitrogen. That is the 50 to 45 milligram per deciliter. It reflects the nitrogenous waste in the blood, that how much non-protein nitrogen like NPN, non-protein nitrogenous waste, is present in the blood. That is the blood urea nitrogen. Estimated GFR indicates the filtration rate of the kidney, that how much kidney is able to filter in a single minute, right? Urine analysis, because urine is also formed by the kidney. Any deficiency or any defective in a portion of the kidney, or ultimately evacuation capacity of the kidney, or presence of abnormal compounds in the kidney which is not normally present in the normal cases like protein, glucose, RBC and more. If any unwanted compound which is necessary for the body and it appears in the urine, then it indicates the inefficiency of the kidney to absorb the necessary compounds. So urine analysis also indicates the kidney defects. Next is creatinine clearance test. It directly measures the kidney filtering rate, that how much kidney is able to filter in a single minute. Electrolyte panel also comes under carefully, like sodium, potassium, chloride, bicarbonate levels. It also indicates the electrolyte balance in the body, because one of the major function of the kidney is the balance of the electrolytes. And by imaging, by ultrasound, by CT scan, we are able to visualize the internal structure of the kidney and to identify the particular disease, right? So they are the kidney function tests which help the physician or which help the pathologist to identify the exact cause of the defects in the kidney. Next is the gastric function test. Next is the gastric function test. Gastric output test. It measures the production of acid. See, in the stomach there is a acid which is necessary for the killing of microorganism, that is the hydrochloric acid. It is produced by the chief cells of the stomach. So this acid is necessary for the digestion, right? So that how much acid is produced, it comes under the gastric function test. Serum gastrin. It is serum gastrin levels is elevated in Zollinger-Ellison disease. Pepsinogen 1 and 2. They are the indicators of gastric mucosal health. Pylori test, urea breath, stool antigen or biopsy. pH monitoring evaluates acid reflex. Schilling test assesses vitamin B12 absorption. Next is the cardiac profile test. In the cardiac profile test, we have troponin, CKMB, myoglobulin, PNP, CRP, ECG, echocardiogram. So these are the following tests we have to assess the functions of the heart. Troponin basically is a kind of cardiac marker which is increased in the myocardial infection, right? It indicates the risk of myocardial infarction. CKMB, that is creatine kinase MB, present in membranes or in the brain. It indicates the cardiac muscle injury, right? Myoglobulin is a protein that is the early indicator of heart muscle damage, and BNP is necessary for assessing the heart function or heart failure sensitivities issue. CRP is a C-reactive protein that is a marker of inflammation in cardiac condition as well as other conditions also, but it is relatively high in the heart conditions. ECG, that detects the arrhythmias or ischemic changes in the heart. Electrocardiogram, or that is the ECG is the electrocardiogram. Next one is the echocardiogram, that shows the structure of the heart and shows the function that is necessary to check the functional capacity of the heart. Next is the infertility profile test. For evaluate the fertility, we have the profile we have FSH, LH, estradiol, progesterone, anti-Mullerian hormone, prolactin, thyroid function test like PSH 3334, semen analysis, HSG, hysto, selfingography, and transvaginal ultrasound. So these are the parameters, so these are the tests by which we are able to check if the couple is fertile or not. So FSH, follicle stimulating hormone, luteinizing hormone, estradiol, progesterone, they are the hormones which are necessary for the evaluation and for the ovarian function. If any of the hormones is below than the normal range, then it indicates that defect in the ovarian function. Next is the AMH, anti-Mullerian hormone. It indicates the ovarian reserve, that how much OVAs or eggs are reserved inside the ovaries, right? Is the eggs production is normal or not? Next is the prolactin. It is also a hormone which is necessary to inhibit uh, high levels may inhibit ovulation. If the prolactin is present in higher levels, then it is responsible for inhibiting the ovulation. And thyroid function tests. See, thyroid function tests are generally performed to check the metabolic capabilities of the body, but directly they are also affects the fertility of the persons. Higher level may result in causing the fertilities. TSH, T3, T4 levels are higher than the normal or lower than the normal result in infertility. Semen analysis by analysis of the semen by microscopic, chemical and physical examination, we are able to measure the sperm count, motility and morphology, by which we are able to detect if the person is fertile or not. HSG, hysterosalpinography, it is necessary for checking of the fallopian tube. Is the fallopian tube is blocked or not? So to check the blockage in the tubes, right, we have the hysterosalpinography, which is a part of radiology department, and transvaginal ultrasound, which is also a part of radiology department, which is necessary for assessing the uterus or ovaries function and to check the follicle development, if the follicles are properly developed or not, because follicles are necessary for the production of the for the ovulation cycle, right? So these are the some organ function test which is present inside your syllabuses. So let's summarize that what I have told you in short. See, first of all, we have learned that what is metabolism and what are the two types of metabolism, anabolism and catabolism. What is ATP, right? Then what is the metabolic efficiency? What is cell? I told you that how energy is produced. What are the helper molecules which help in the production of the energy? That how the nutrients will broke down to produce energy. What is glucose breakdown? What is your fatty acids breakdown? What is your amino acids breakdown? Right? Then we will discuss about the TCA cycle, right? Then about the electron transport chain, that we saw that uh, which energy, which biomolecule have the higher energy, fat or the carbohydrates. Then I told you about the organ function test. What is the organ function test? Why the organ function tests are necessary? How many types of organ function tests we have? In which we studied about the LFTs, right? We studied about the KFT. We studied the gastric function test. We studied the cardiac profile, infertility profile. So this is all about the organ function test, right? One more thing, that every biomolecule have a different metabolic pathway, right? Different types of the anabolism and catabolism pathway. Collectively, they have the different metabolic pathways to produce the energy, right? The goal of each and every product, each and every biomolecule is the production of energy or to synthesize the new products for the body. So each and every biomolecule is finally related to the production of energy, है ना? And for the production of new compounds like cell division, cell growth. So see, I just explained you. I just give you an example. Like when you are infant, your size is small, your bone size is small, your brain size is small, your organs are small. But when we grow day by day, our size, our body mass will increases day by day. So ultimately, what happens? The cells are dividing. The cells are dividing, and they will increase the size. And ultimately, the division of cells is performed by the energy. If the body has doesn't, if the body doesn't optimum amount of energy and acquired amount of energy, then the cells will not divide and the growth of the body doesn't take place. So for each and every growth and for every process, like for the growth, body growth, for the development of cells, for the production, for the reproduction, what is required, the energy is required. And from where the energy is come, the energy is come from the diet we take. So the diet we take is finally goes down into the body, and it is break down into the smaller units, and finally the smaller units are absorbed by the body, by the small intestine, and then the absorbed smaller units is goes down into the cell, by crossing the cell membrane, they enters the cell, and then inside the cell, the glycolysis, the glucose, glycerol acid or fatty acid breakdown, the amino acid breakdown will take place and leads to the production of what? Leads to the production of ATP, that is in the form of energy, right? So it is all about the overview of biochemistry metabolism. Thank you so much, students. The detailed unit-wise videos are available on your LMS. If you have any doubt, then you can write down in the chat box. I will answer your queries. Thank you so much, students.